Let us discuss the third type of carbohydrates today, that is polysaccharides. Till now we have studied the two types of carbohydrates, monosaccharides and disaccharides. Today we will discuss with the third type of carbohydrate, that is polysaccharides. Now polysaccharides are the carbohydrates which are made up of many sugar units, or they are made up of thousands of sugar units. Thousands of sugar units, they come together, they join together and then they form polysaccharide. Okay, now this many monosaccharides actually they come together and they get added with each other one after another and they form a long chain which forms a large molecule, that is which forms a polysaccharide. This process of formation of this polysaccharide is called polymerization. This process is called polymerization. This is written here, the polymerization. Many monosaccharides they get added with each other one after another, forming a long chain which ultimately forms large molecules or polysaccharides. This process is called polymerization. Now these polysaccharides they are broken down into monosaccharides by hydrolysis process. That is, it reacts with water and then it gets broken down into monosaccharides, into monosaccharide units. So polysaccharides they are broken down into monosaccharides with the help of hydrolysis process. Properties of this polysaccharide, it depends on the properties of polysaccharides. It depends on its length, as it forms a long chain. So how long it is, based on the length of the polysaccharide, their properties depend. Their properties also depend on the branching, whether the polysaccharides they are branched, their structure is branched or not, and whether the structure is having folds and coiling. So based on the folding and coiling of the structure of this polysaccharides, the properties are depending. So properties of polysaccharides generally depend on three things: its length, branching, folding and coiling structure. Further, this polysaccharides they are classified into two ways. There are two methods of classification. First, the classification of polysaccharide is based on monounits. It is based on monounits. Based on monounits, polysaccharides they are divided into two types: Homo Polysaccharides and Heteropolysaccharides. Homo Polysaccharides and Heteropolysaccharides. So Homo Polysaccharides, the example is cellulose. So cellulose is made up of only glucose molecules. Many glucose molecules they come together and they form cellulose. So there is only one unit, one sugar, that is glucose, right? So only glucose forms. So it's a monounit. Only one glucose, only one sugar forms cellulose. So cellulose is an example of Homo Polysaccharides. It is a Homo Polysaccharide because it is made up of only monounit of sugar. Whereas Heteropolysaccharide is made up of different sugars. Different sugars they come together and they form the Heteropolysaccharides. Example, hemicellulose. Example is hemicellulose. So like hemicellulose is formed of fructose, xylose and mannose and so on. So here there is fructose, there is xylose, there is mannose. So different sugars are come together and they form the heteropolysaccharides, and that is hemicellulose, and that is why it is the heteropolysaccharides. So this is the first method of classification of polysaccharides based on the monounits. Which units come together and form the polysaccharides, based on that they are divided into Homo Polysaccharides and Heteropolysaccharides. The second method of classification of polysaccharide is based on their function or their role. Based on the function or their role. Based on their function, they are divided into two: storage polysaccharide and structural polysaccharide. Storage polysaccharide and structural polysaccharide. Example we will see storage polysaccharide. The example is starch. We know that plants they produce the food in the form of glucose, but excess glucose gets converted into starch and gets stored in the storage organs of the plants, like roots, fruits, seeds. So starch gets stored. So starch is a storage polysaccharide. Starch is a storage polysaccharide. Some of the polysaccharides they are called structural polysaccharides because they help in the formation of structure of the cell. For example, cellulose. We know the cellulose it helps in the formation of cell wall in plants. So it helps to form the structure of the cell, in the structures of the cell. So it is called structural polysaccharide. Okay, structural polysaccharide. Example, cellulose, which helps in the formation of cell wall in the plant. So based on the function, the polysaccharides are classified into two types: storage polysaccharide and structural polysaccharide. So now let us discuss the examples of polysaccharide. The first example which we are going to discuss of polysaccharide is starch. The first example which we are going to discuss for polysaccharide is starch. So starch is a stored food in plants. Now plants, they produce food in the form of glucose and they utilize it as soon as it is produced. But the excess glucose which is present in the plants, it is converted into starch and it gets stored in the plant parts. Storage organs like roots, fruits, seeds. In such organs, the excess glucose or the excess food gets stored in plant parts. So that gets stored in the form of starch. And that is why starch is a storage polysaccharide. That is why starch is a storage polysaccharide. If we see, starch is made up of only glucose molecules. Well, it is made up of monounit. Only glucose molecules, they come together and they form the starch. As only it is made up of only one molecule, that is one molecule means only one unit, that is only glucose molecules, they come together, many glucose molecules, they come together and they form starch. And that is why this starch is a homosaccharide, Homo Polysaccharide. Starch is a Homo Polysaccharide. We discussed previously about the classification of polysaccharides. So based on monounit and based on the function. So based on the monounit, starch is Homo Polysaccharide because it is made up of only one unit, monounit, that is all sugar molecules. And according to the function, starch is storage polysaccharide because it gets stored in plants. Now starch mainly consists of it exists in two forms. Starch mainly exists in two forms. One is amylose and amylopectin. Starch exists in two forms, amylose and amylopectin. So amylose means what? Then amylose, many glucose molecules they come together and they form amylose. If we see, the number of glucose in amylose is 200 to 2000. 200 to 2000 glucose molecules, they come together and they form amylose. Okay. So when these glucose molecules, 200 to 2000 glucose molecules, when they come and join with each other, obviously they will form bond with each other. So when two glucose molecules form the bond between each other, the bond formed is 1,4-glycosidic bond. We have discussed 1,4-glycosidic bond. When one glucose molecule will form bond with the second glucose molecule, the bond formed is 1,4-glycosidic bond. That we'll see in detail. If you see, this is the structure of amylose. So this is one glucose molecule, this is the second glucose molecule, this is the ring structure. We have studied it's a pyranose ring, which consists, it's a hexagonal ring of glucose. There is one oxygen present in it, one carbon here, the second carbon here in the ring, third carbon here in the ring, fourth carbon, fifth carbon on the ring, and sixth carbon is extended outside. It is branched outside. Okay, so it is not on the ring, but it is outside, that we have discussed. This is one glucose molecule. The second glucose molecule we have made over here. Again, it contains one oxygen. First glucose, second, third. First carbon is here, second carbon here, third, fourth carbon, fifth carbon, and sixth carbon is branched outside. So the bond formed is between the first carbon and the fourth carbon of second glucose. First carbon of first glucose and the fourth carbon of second glucose. That is why this bond formed between two glucose molecules is 1,4-glycosidic bond. Whenever we talk about 1,4-glycosidic bond, you understand that it always forms a linear structure, straight chain. It will form always a straight chain. Okay, linear structure. And that is why we say that amylose, it has linear structure, but at the same time it has coiled structure. Coiled means like spring, how the spring is coiled. Though it's a linear structure, but it is not exactly straight, but it is coiled structure. And that is why we say that amylose shows linear and coiled structure. This is amylose. The second part of glucose, or in the second form in which starch exists, is amylopectin. Now in amylopectin, if you see, the number of glucose molecule is 2000 to 20,000. The number of glucose molecules in amylopectin is 2000 to 20,000. Here the number of glucose is more. Again, when the glucose molecules, they come together, they form the bond. The bond formation in amylopectin is 1,4 and 1,6 glycosidic bond. 1,4 and 1,6 glycosidic bond is formed in amylopectin. So that also we will discuss. This is again the glucose molecule, first glucose molecule, second glucose molecule, like in amylose we discussed. So then first the 1 and 4 glycosidic bond is formed. Just now I have explained you this. So in 2 glucose molecule, 1,4 glycosidic bond is formed. When we talk about 1,6 glycosidic bond, then you see the 6th carbon is outside here, branched. Okay, not on the ring, but it is outside. And if we draw another glucose molecule over here, oxygen is here, first glucose is here, first carbon is over here, second carbon is here, third, fourth, fifth, and this is 6th carbon again outside. So when we see this first carbon of this glucose forms bond with 6th carbon of this glucose. So this is called the 1,6 glycosidic bond. This is called 1,6 glycosidic bond. So whenever we, as just now I told you that whenever we talk about 1,4 glycosidic bond, it is a linear structure. But when we talk about 1,6 glycosidic bond, it starts forming branches. Whenever glucose molecules they form 1,6 glycosidic bond, it starts forming branches, and that is why amylopectin shows branch structure. It shows branch structure. Now when we talk about this starch, this amylose and amylopectin, they are present in the ratio of 1:3. Amylose and amylopectin, they are present in the ratio of 1:3 in starch. And when starch reacts with iodine, it gives blue-black color. When starch reacts with iodine, it gives blue-black color. That blue-black color is because of amylose. Okay, as amylose is present in starch, and starch, when it reacts with iodine, it gives blue-black color. That blue-black color is mainly because of amylose, which is present in the starch.